morning, everyone. Our next speaker is Rebecca Asch from Princeton University, and she's going to be talking about climate change and trophic mismatches between plankton blooms and fish.
Okay, thank you everyone for sticking until the last day of the conference for the most part. Thank you to the organizers for inviting me here today. So as Jasmine said, I'm going to be talking about how climate change likely affects mismatches between plankton blooms and the timing of when fish spawn. And throughout this talk, I'm going to be using the word phenology quite a lot. So I just want to make sure that everyone's on the same page and knows what this means. So phenology refers to the study of seasonal biological cycles and how they affect climate and weather. So the slide here has a number of examples of different phenological events. So this is bud burst, where plants have buds that come out in the spring. For animals, the two types of phenology that are most often studied are either migrational phenology or reproductive. So monarch butterfly migrations, salmon migrations, and for reproductive phenology, which is the focus of this talk, I have an example of a cute baby seabird here. So a question that you might be asking is, why is phenology of interest to fisheries oceanographers and perhaps to biological oceanographers in general? And for me, the answer of this really stems from the fact that many fish species have very high fecundity, but in a stable situation where populations are at equilibrium, only 2 of potentially millions of eggs will survive, so survival rates are very low. And that means that very small changes in those survival rates can be important for population dynamics. And this is an example here for haddock, an important fish species here off the northeast coast. This is showing you a time series of recruitment. So for people who are not familiar with fisheries, recruitment is the number of young fish entering a cohort where they become accessible to fisheries. And the point I want to make here is it's not unusual for recruitment to vary between years by several orders of magnitude. And a lot of that variation is really driven by basically oceanic conditions, including seasonality that can influence survival of eggs and larvae. So there's many hypotheses out there that connect recruitment variability to oceanic conditions. Since I'm focusing on seasonal aspects, I'm going to be talking to you today about the match-mismatch hypothesis. This has been around since the 1970s. It was developed by David Cushing, although there are some aspects of it that go back further to the work of Johann Herc. And this hypothesis basically proposes that many fish species have evolved to spawn at about the same time as peak plankton production in order to maximize the survival of their offspring. However, there's years where conditions look more like this, and you get mismatches between the timing of plankton and fish, and those years are often characterized by poor larval survival, slower growth, and this can result in less recruitment and then impacts on fisheries. So why can't fish consistently reproduce at the same time as plankton blooms? And the reason for this is actually pretty simple. Fish can't predict the future, but octopuses might be able to predict World Cup outcomes. So the reason why fish can't predict the future is that many fish species actually prepare to spawn over a course of several months where they build up gonadal biomass. And the graph down here at the bottom is showing this. This is the gonadal somatic index, which is basically the percentage of fish biomass that's made out of gonadal material. And it increases over time, often as a function of temperature measured in degree days. So this means that basically, since fish are poikilotherms, that under many circumstances, their metabolism basically is accelerated by warmer conditions. Their gonadal biomass builds up more quickly and they spawn earlier when it's warm. And this is a meta-analysis that appeared a couple years ago in Nature Climate Change that kind of shows you that this actually does happen in reality, and it's common among many species, not just fish. So this is showing you different organisms here versus rates of phenological change from long-term time series. And what you can see is the majority of things are getting earlier as temperatures warm. I want to highlight a couple aspects of this figure. So circling phytoplankton and larval fish with zooplankton in the middle. So both of them are showing advances in phenology, but the rates aren't the same. And based on this meta-analysis, it actually looks like fish might be changing more rapidly. So we might get more mismatches in the future. I also want to point out that for this meta-analysis, there's only 2 data sets included on fish. So we're dealing with small sample sizes, which could lead to uncertainty. Fortunately, there's been some new studies of this since 2013. So one of them is work that I published last year. This came out in PNAS, and this is based on cow coffee data analyzed decadally. So I looked at the phenology of 43 larval fish species, and this is its first principal component, and there was a trend for basically phenological advancement there. This is zooplankton data from cow coffee as well, and here you see there's a lot of variability between decades, but not really a trend. So again, suggesting that there might be differences in how trophic levels are responding to climate change. This is a study that actually just occurred in Nature a couple of weeks ago, and they found contradictory influences on phenology, often of temperature and precipitation in terrestrial environments, but they also included some data from marine and aquatic habitats. And when they have summed across different influences, they found that changes in phytoplankton actually sum out to be fairly low. And then for higher trophic levels, crustaceans and fish, the changes are actually projected to be quite rapid. So again, this is suggesting we might get more mismatches in the future. I'm going to be exploring this in greater depth during this talk based on some work I've been doing at Princeton, and this is going to be based on Earth system models. So this work is based on 3 hypotheses. The first one is rather evident given my introduction. So one would expect that under a climate change scenario, we might expect fish to spawn earlier and phytoplankton blooms to occur earlier. However, we might not expect these to occur at the same rates, so we might get more mismatches. And the reason why we won't necessarily expect them to occur at the same rates is that often the drivers are different. I've been emphasizing here that temperature is probably the predominant driver for fish, where for phytoplankton, we know the bloom time is a complex interplay between nutrients, between light limitation, between predation, and temperature is one factor among many. The other hypothesis I'm going to be exploring here, which I'm kind of excited about, is how range shifts and phenology interact. And we know that from the last talk that many species have experienced shifts and shuffle, and that their ranges are moving poleward. And this is a way of basically tracking climate change and climate velocity. So this might allow basically organisms to kind of adapt and diminish the amount of mismatches between trophic levels. So the model that I'm working with is the NOAA Geophysical Fluid Dynamics Lab, or GFDL, Earth System Model. This is one of the ones included in the last IPCC assessment report. And its ocean ecology and biogeochemistry component is basically a nutrient phytoplankton, zooplankton detritus model, and it's shown here. There's 3 functional groups of phytoplankton. For the purpose of this talk, I'm summing across them and looking at kind of chlorophyll as an indicator. And you might be asking why I'm focusing on phytoplankton. And in this community, we know that phytoplankton are of interest in and of themselves, so changes in their phenology affect annual primary production and potentially at times export production. There's also a good connection between phytoplankton phenology and fish. So this is an example that was published over 10 years ago that I think at this point is somewhat of a classic example, looking at timing of blooms and then survival of haddock. The other reason, which is actually probably the biggest motivation, is that I can validate the model using ocean color data, where for zooplankton, there's about 20 time series of their phenology shown here from work by Macus. And you can see that the spatial coverage is exclusively northern hemisphere and also rather coastal, often in inland seas. So we just don't have global coverage to check to see if the model's working well. So in terms of how I detected the timing of phytoplankton blooms, this algorithm was designed so that it would identify blooms in every pixel of the model in every year. So it basically has a threshold that's specific to a pixel and specific to the year, which is the 75th quantile of the annual range, and blooms start at that point. They end when the pixel drops below that, its time series drops below that threshold, and it needs to stay there for 5 time steps so that I'm sure that this is a seasonal process rather than subseasonal variability. From this, I can look at other metrics such as bloom midpoint, duration, and magnitude. I'm focusing here on just the first bloom of the year, although I actually do have data on multiple blooms per year in some regions. This is going to be based on the RCP 8.5 emission scenario and going from pre-industrial to the end of the 20th century. So how well does the model work? This is information from the model on bloom initiation dates shown in different colors. This is from CWIFs, and these are both 10-year climatologies. So the spatial correlation is, okay, here's my pointer, about 0.6. And if you look at other metrics, the midpoint and end of the bloom, they're similar. The model does a little bit better when looking at the size of the bloom. It doesn't do as well when looking at bloom duration. In terms of biogeographical patterns, the model does show basically spring blooms in subpolar gyres that get later into the year, going up to high latitudes, and in the subtropics, we tend to have more fall and winter peaks in chlorophyll associated with mixing. I think the one other thing just to mention here is you can see I'm not looking at the Southern Ocean because we know that there are some model biases there due to basically mixed layer depth that influence timing of phenology there. So you see, since this talk is a theme on marine thresholds and regime shifts, this relates more to thresholds, and I want to do actually some Bayesian change point analysis just to connect that to the theme. So this is the algorithm that I've been using and a couple of example time series from individual pixels. So these dashed lines are the change points. So change points are very commonly detected among phytoplankton bloom initiation dates. A lot of the change points were changes in the slope. So you have a mean with variability, and all of a sudden, most of these time series, you get kind of at some point a decreasing trend, so the slope changes, although the dates of that can be quite variable between pixels, and these are two that are located near each other. There are less commonly changes in the mean, so you have high variability around one mean, high variability around another mean, a very abrupt shift, maybe a tipping point in between them. So those were individual pixels. This is averaged data zonally to give you a better sense of spatial patterns. So what you want to see here is that once the data are averaged out, you do see this decline in bloom timing where it gets earlier in the year across pretty much all latitudes north of 40 degrees. And this often starts very early. So at high latitudes, you can see it happening beginning kind of in the late 20th century. And later on, I'm going to be talking about a baseline period versus a future period. My baseline is 1901 to 1950, and that's why that rather early baseline was selected. Once you move into the subtropics and tropics, I found that there's a lot of very high variability in plankton phenology, but not really as much of a climate change signal. So the rest of this talk on comparing data against fish is going to be focusing on these areas where you really see a very clear signal in phenological change. The other thing that I just want to mention here is that basically, in addition to looking at changes in timing, mismatches are going to be moderated by the length of the bloom and its magnitude, because that will also influence the amount of overlap between trophic levels and how much food is available. And in general, we see that blooms are getting smaller and that often they're getting shorter, although growing seasons could be longer because I'm only looking at spring blooms and we are seeing increases in fall blooms in some areas. Not going to dwell too much on the mechanisms behind bloom timing because I know last year at this workshop there was a whole session focusing on this, and it's probably been covered in great depth. But there's a lot of hypotheses out there, and one of the things that they all have in common is that they relate to processes relating to mixing and stratification. And in the model, there is a good correlation between bloom timing and stratification between 0 and 100 meters. So this is the R square for that. In all areas except for where you see that stifling up here, you have basically negative correlation. So blooms are earlier when stratification is greater. You can partition the changes in stratification to the effects due to temperature and salinity, and that's what I've done using an approach developed by Caputundi. And in terms of the changes we're seeing, while temperature is important in some areas, you can actually see that stratification is often driven by changes in salinity and freshwater input, which makes sense given some of the earlier talks that we had about the Arctic earlier today. The magnitude of the scale bar is actually twice that of this. So that's really emphasizing that this is not just a temperature effect and that there are multiple oceanographic factors that affect bloom timing. So I'm going to be getting into fish right now, and I'm mainly focusing on temperate epipelagic fish. And in terms of modeling them, I'm not looking at specific species, but I have two groups of species to basically provide different behavioral types. So I have geographic spawners represented here by these fish in seagrass habitat in that cartoon. So these are fish that will only spawn in the same place regardless of environmental conditions. So salmon would be an extreme example. In contrast, we have these fish that are world travelers, that their spawning grounds may shift depending on environmental conditions. So as a starting point for this model, I'm assuming there's a baseline period where on average there's a match between phytoplankton, the start of the bloom, and when fish spawn. But we also have interannual variability, even that baseline period, and interannual variability is controlled by degree days, such that basically warmer temperatures result in earlier spawning. I'm assuming, although this is an assumption that I can relax, that right now there's not any genetic or behavioral adaptation, so there's a threshold where you have enough degree days accumulated for spawning, and if there's no change in that threshold in the future, one can project forward basically when fish would spawn in my future period. So how do the rates of change compare among phytoplankton and the geographic spawners? So this is the data that I've already shown you for phytoplankton. It's a histogram showing the amount of change between the two time periods I'm looking at. And the median rate is about 17 days, so about 2.5 weeks of change in bloom timing. This is the equivalent histogram for the geographic spawners, the fish that have fixed spawning grounds. And they're predicted to have much larger changes in their phenology. And as you might guess, that results in mismatches, and that's what's shown here spatially in this map. So the red areas are where the fish spawn before the bloom, the blue is where they've spawned after the bloom has ended, and white is coincident with bloom. These percentages are the amount of study area that falls into each category. So across 86% of my study area, the fish are spawning before the bloom. Of course, that might be moderated by movement and range shifts. So how did I look at range shifts among environmental spawners? The way that this was studied was basically, I assumed that these fish had a preferred thermal habitat. There was a 2-degree band of annual isotherms. So in this case, this fish prefers this green area, which is between 6 and 8 degrees, and that they'll move to basically track changes in those isotherm bands. So this fish would end up moving from here to a bit northward and westward to stay in that same habitat. So now I'm going to be comparing the geographic and environmental spawners. So this is the map that you've seen beforehand for the geographic spawners, and here's the equivalent map for environmental spawners. And you can see that there's a lot of white here, so there's a lot more areas with matches between these trophic levels. And basically, beforehand, we had matches in 10% of the study area. Now that fish can shift their range, we get up to 60%. So that's a better scenario. The other thing that I'm going to kind of talk about before opening up to questions are extreme events. And in this context, I'm considering an extreme event to be a mismatch that's larger than 1 month. That is somewhat of an arbitrary number, but the reason why I picked that is most fish have egg stages that last a couple days, and then another couple of days where they have yolk sacs that they use to feed on. A month is substantially larger than the time that you would think that those two periods would add up to for a typical life history. So this is kind of a point where a fish would definitely be in need of abundant plankton resources. So these are changes in areas with extreme mismatches for the Atlantic and Pacific and for geographic and environmental spawners. For the environmental spawners, you can see that there's intraannual variability, but not much trends. For the Atlantic and Pacific geographic spawners, there's a very pronounced increase where these fish are experiencing more and more mismatches that are quite large over a month, getting up to close 40–50% of the study area experiencing this in any particular year. This is a map of the same data to show you the spatial patterns, and it's looking at the ratios between future and baseline probabilities in experiencing an extreme mismatch. So ratio 2 means that basically these mismatches have doubled in their frequency, where a ratio of 0.5 indicates that they've been reduced by 50%. And although the environmental spawners, their time series look like they were very steady, there's a lot of spatial variability, and there are areas where you do start getting more extreme events, where this occurs quite frequently among the geographic spawners, and there's lots of areas where basically the frequency of these extreme mismatches increases tenfold. So returning to my hypotheses, yes, I did see that both phytoplankton blooms and fish spawning got earlier. I did see an increase in the frequency of seasonal mismatches, although I label this as most likely because it's dependent on some assumptions I made about how to model the system. In terms of range shifts, I did find that they ameliorated the extent of seasonal mismatches, although there was some spatial heterogeneity. So in terms of the broader implications of this is really suggesting that species that are tied to specific habitats might be the losers under climate change, or ones that can undergo range shifts. This might actually be a good way of adapting to changes, and they might flourish, but in different areas. The other thing that I really want to highlight, which is nice because it was a point that Phil pointed out earlier in his talk, is that climate variability is quite important. And in this case, there's been very little attention given to how changes in extreme events might affect ecosystems. So I think this is an area that we could, that needs more consideration and study in the future. And lastly, I want to emphasize that this influence of degree days on organismal phenology is common to a wide variety of organisms, from plants to mammals to fish. So this approach is something that basically one could adapt to whatever your favorite species is, potentially. With that, I'll see if there's time for questions.
Hi, Rebecca. I'm just wondering about one of your earliest slides when you showed some of the phenological changes proposed from other studies for zooplankton and crustacea, and that scale was close to the larval fishes and other fishes. So wouldn't you expect that the mismatch would be occurring possibly between phytoplankton and zooplankton instead of for larval fish?
Yeah, that's something that is a really great question, something that I'd actually like to look at in the future. And let me see, I think at one point I highlighted that paper by Dave Nakas, and he has done a really thorough analysis of zooplankton phenology from a lot of sites, and he has found very large changes in their phenology related to both climate variability and, in some cases, change. And he argues there, although he doesn't have any fish data in that paper, that it might be larger than the variability in fishes. So that's definitely something that's, I think, is quite a possibility. And I do want to actually look at this with Cobalt, our second GFDL model that has more details on functional groups of zooplankton. When I started this study, that data were not available to look at on a kind of a sub-monthly basis. They are now, so that's something I want to follow up on. And I think that the question there is also just, there might not be mismatches between zooplankton and fish, but what Mac has found in that paper is that the phytoplankton phenology and zooplankton phenology weren't tracking each other. So this might mean that even if there's not mismatches between fish and zooplankton, if there's mismatches between zooplankton and phytoplankton, that could affect zooplankton production and then fish. So that's a question that I'm hoping to get to in a couple of years.
That was a great approach. I have one question. You know, there are certain areas, we heard some yesterday in the Indian Ocean, Arabian Sea, and of course you could think of the Gulf of Mexico, where because of the shape of the basin, fish can't migrate any farther north. They're capped. And so if things warm up there, those would be kind of, and others, interesting places to look at.
Actually, I'm on a proposal right now to, I think, the NOAA Restore project in the Gulf of Mexico, where part of our aim is to look exactly at that. It's looking at spawning aggregations of reef fish, and one of the reasons why that's an interesting area is because that is an area where you have basically the coastline is running east-west. So that's something I'm definitely interested in. I think Malin Pinsky has done a little work on that in his paper, and he's found that in those regions you do have shifts in fish distribution, but it's east-west, and that they are tracking climate velocity, but they're constrained in how they can do that. So that's why I would hypothesize would probably happen there.
Hi, could you comment on the potential for selection in spawning and whether there's a sense of how much plasticity there is among fish populations?
Yeah, so that's a really good question. And one thing that we know is that, so I'm emphasizing temperature here, but there's a lot of other things that can influence spawning time. So in salmon, there's a lot of adaptation by different stocks, and that actually seems like the local adaptation for salmon is the predominant factor for that particular subspecies. That hasn't been found among other fish, but I think one thing that's really important that might get to this is that there are, that basically size of fish and population structure can really impact phenology quite a lot, which suggests that there is kind of potential for plasticity there. And as I have said, that it's something where I think can be incorporated into this model, but it's something where I would need to be able to figure out what that rate of change is and parameterize it. I think at this point we don't have necessarily the data there, but I have ideas of if anyone knows what that rate should be, how I could parameterize it and kind of loosen up that constraint of no genetic evolution.
Any more questions for Rebecca? Okay.
Hi, Jim Bishop, UC Berkeley. On Monday, I showed the very curious feature of low biomass and very high export. And also in 2009, I published a paper in the Southern Ocean in Biogeochemical Cycles where the only interpretation of the data from a year-long time series of floats in a developed 400-meter mixed layer, one observed a bloom and one didn't. And the only way that and one observed high export to depth and one didn't. And the one that had the bloom had little export. The one that didn't have the bloom had high export. And the only way I could tie that was to the fact that the float sampled the diurnal cycle of stratification and detected 0.01 sigma theta stratification events prior to seasonal stratification. So in that paper, it seemed that the bloom timing was in fact determined by the food web response to transient stratifications before seasonal stratification. So is that part of the explanation for mismatches that you talked about in your presentation?
So I guess one question, and this is, I wasn't here on Monday, is in terms of food web timing, was it focusing on microzooplankton versus macrozooplankton, just because we have a couple different food webs here, and just understanding the study that you did might be important for answering that question.
Well, we just deployed floats with sediment traps that recorded sedimentation at hourly frequency at various depths in the water column to 500 meters. And we were in the Santa Cruz Basin in the California borderlands. And in that case, we found the highest sedimentation fluxes in the month of January, whereas the later timing, you would say May, the waters are much more biomass rich. So the whole issue was that we found a much tighter coupling between production and export. And in fact, the whole ecosystem was spun up. There were seabirds, there were marine mammals, there were squid. The whole water column was alive with biology in January. And then in the case of the paper in Global Biogeochemical Cycles, this was just simply two profiling floats measuring the hydrography and biomass variability and an index of sedimentation, which gave us the interpretation of no bloom, better food web coupling of the zooplankton to the community of phytoplankton, so that they're grown up ready to harvest the phytoplankton at the rate that when permanent stratification kicks in. So it's kind of related to the
Yeah,
depth.
I think that's a good question. I guess the reason why I asked is my instinctual thoughts about that are that it might depend on different members of the zooplankton community, because certain species can be much more reactive to changes and the coupling would be tighter. So that's obviously going to be the case among microzooplankton, whereas if you're in a system where diapause is quite important, then there might not be as close coupling with kind of copepods and zooplankton. So that's kind of, I think it might have to do a little bit with community composition. But I think that's a really interesting set of observations, and happy to talk more about that.
Okay, let's thank Rebecca, and I think we have a break now.